Proteomics is an important branch of molecular biology. And in this module, we will discuss the analytical proteomics, and particularly we will focus on the methods that how we can separate the proteins. One of the best separation tools which are available is two-dimensional gel electrophoresis, which was invented in 1970s. That has been extremely used for separating the different proteins. You can see that an average 2D gel, it can resolve only about 2000 proteins. When there are 2000 proteins, less than 2000 proteins present, the 2D gel electrophoresis can separate those proteins into the different segments. For example, in this photograph, you will see that there is one short one-dimensional electrophoresis, then there is a long one-direction page or electrophoresis, and then on the extreme is a 2D page electrophoresis. You can see that the protein fractions which are separated through one-dimensional and two-dimensional SDG page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis technique. Now, one is very short band which are present, but on the 2D, the one band is actually separated into the more protein bands, giving us a better separation. And the one band, you can see, has been spreading more. It has given us the more bands. So this is the important technique for separating the proteins. But there is a problem with this method as well, because many interesting membrane proteins, they are too hydrophobic to dissolve in the buffer which are used in two-dimensional electrophoresis. And definitely when these proteins, they are not soluble, they won't be shown in the 2D electrophoresis. And they cannot be seen at all. So if those membrane proteins we try to do with the 2D electrophoresis, we cannot see all of them. We cannot see all bands which are present there. So how we will do this? Because many proteins, they are present in such tiny quantities in the cell that a 2D gel, it cannot detect them. That's another problem. One problem is that they are hydrophobic. They are not dissolved, all of them. And the second is that they are present in very tiny amount, which 2D gel cannot detect them eventually. So most of these problems are presently interactable. That is, those problems, they are interacting with each other, and we have to overcome in order to make a better separation. Now, scientists, they dealt with 2D gel resolution problem by analyzing the different cellular compartments separately. So what the solution they found, that they took the different cellular compartments separately rather than taking a whole. They took one tiny part of a cell, then the other part of the cell, and make that the analysis. So a subcompartment like the nucleus or of a protein assembly, like the nuclear pore complex, are the good example of understanding this concept. That is, that they use a subcompartment in order to analyze the proteins present in that compartment, because 2D cannot detect the tiny amounts present over there. And that is how scientists overcome that. The problem which we found is that they are the hydrophobic proteins. They cannot be detected by the 2D gel electrophoresis. And the second one is that they cannot detect the tiny amounts which are present. And in that case, we have to analyze, or we have to divide that in the subcompartment, like the nucleus of the protein assembly, like the nuclear pore complex which are present in the cell, and we solubilize them, and then we detect the protein which are present in the nuclear pore, that is in the one compartment of a cell, through the 2D gel electrophoresis.